for me an absolute privilege to be able to provide you with a brief overview of the ongoing research of the Macadamia Protection Program at FABI. So as you'll see, we're doing things a little different this year. Luckily, it's not just me. We have divided the talk into a number of short talks. I will give you a brief overview of our research program. I'll talk about the disease diagnostic clinic. I'll then hand over to Angel that will talk about Botrytis branch dieback. Dee will talk about husk rot, and we'll end up with Ashley and Elisa that will talk about macadamia nut borer as well as stink bugs. But please also come and visit us outside. We have a stand, and if you have any interest in macadamia fingerprinting, wanting to know anything about genetics and genomics of macadamia, please come and visit Mary and Melissa. They're from Forest Molecular Genetics, also at FABI, so please come and talk to them. So when we think about the research and what is our overall aim, then we can often divide it into different research focus areas. Everything that we do, we think we want to deliver value, and so we want to think solution-orientated. But from time to time, we have to conduct surveys. Why? I think it's important for us to confirm that we actually know the causal agent. When you think about a pest, do we know that what was dominant a number of years ago is still dominant today? But then we have a strong focus where we want to focus on the biology. So it's about understanding the pest and the pathogen in its environment and the interaction with the host. So why do we do that? That can often help us to really time an intervention. And then when we think about intervention, we have some in vitro screening processes that's ongoing, but when we think specifically about our pests, there's a strong focus on the development or the improvement of biological control and specifically pheromone-based products. Then in addition to research, we also offer services to industry. One that I hope many of you are familiar with is our disease diagnostic clinic. So in terms of this, it is about us monitoring what we actually know, but maybe more important is to ensure the early identification of new pests and diseases. And this then often translates into new research focus areas. And for this, I would like to briefly highlight the polyphagous shuttle borer, so bark and ambrosia beetles, and then I'll touch on dry flower. So when we think about bark and ambrosia beetles, let's quickly recap the polyphagous shuttle borer and macadamia. So remember, we first picked it up in 2019 in macadamia. Remember the study that we did? It was a pathogenicity trial where we tested the fungal symbiont against macadamia. And what we found is that fungal symbiont, Fusarium ulacea, actually grows very slowly in macadamia. So at the time, we hypothesized that when we think about macadamia, we don't expect to really often see mass attack of the beetle, and the beetle killing the tree, which is then a combination of the beetle and Fusarium dieback. And so when we look at our recent monitoring that we did, what were the numbers? Well, if you think about it, it was 12 traps over 3 months per province. Our numbers remained low. Remember, the traps were also set out in places where we know the beetle is present. So for now, our current hypotheses are still standing. But what did we see in high numbers? First of all, we saw 2 species, but species that we do not regard as economically important. What we want to perhaps look for in the future would be Xylosandrus species, Ambrosiodium species. Those are ones that we've also previously found on diseased nuts, and that will be then our next focus when we do another 3-month survey early next year. Then the final example for me, and thinking about the value of the clinic, is dry flower. So we first picked it up in 2018 on a single farm in Lavubu. At the time, nobody thought it's something of importance. But I think since then, of course, it has changed. So the following are 2 tables. This is recent surveys that was done in Australia. And if you can see, there's a number of new Pestalotiopsis species that we associate with dry flower, a number of species that we associate with green mold. What did we find, or perhaps more Gerard in his surveys? A number of species we see in Australia is also present in South Africa, but in actual fact, we have a number of unique species. So now that we've done a survey, what do we know about its biology? Again, we can look at some research that was done in Australia. It's a PhD student of FABI. And what he did is he specifically went and he calculated what is the optimum growth and sporulation temperatures and relative humidities of what we say these different flower bite fungi is. But what does it mean, and is it of value? Well, what we did is we collaborated with Ilaria from the ICFR, and she helped us to construct a risk map. So she added this data onto her macadamia site map. And perhaps what I just briefly want to show you today is what you can see when you look at the risk between new pest Latiopsis, in other words, dry flower, versus green mold. So you would see lots more red and purple on the one side if we talk about risk. And that just tells us that, yes, this is something that perhaps is of more economic importance than what green mold would be. And that also lines up with the research from Gerda, where most of what he found during his survey was in actual fact dry flower. But then what else, and what else are we doing in terms of the biology of this fungal group? That links to Byron's PhD. First of all, we're interested in understanding disease progression. So when you think about healthy leaves and healthy flowers, how does that compare, and what do we see present versus diseased? But also thinking in terms of the old racemes or yellow halo leaf blight, could that be a source of inoculum? And that is sort of work that's really going to complement the ongoing work of Marietta Skuman from the ARC that's looking at to understanding spore loads in the orchard. And then finally for Byron, we also want to develop a rapid diagnostic. It's something that you can actually use in field, because being able to confirm the disease and then respond quicker is what we also see as valuable.
Good morning, ladies and gentlemen. My name is Angel. Thank you, Gerda. I'm going to briefly talk to you about the Botrytis virea in the macadamia canopy. What do we know about the Botrytis virea? So we know that it's causing one of the most commonly reported disease, branch dieback, which is characterized by tip dieback, bark discoloration, and in severe cases, tree mortality or tree decline. This disease is caused by a group of fungi called the Botrytis viriaceae, and they exist as either saprophytes, feeding on dead plant materials, as endophytes on healthy, and as pathogens. So to understand this disease, in South Africa, we compared between both healthy and diseased trees across different regions in South Africa. And what we see there is that we got several species that were found occurring on both healthy and diseased overlapping. And we found that on healthy we got only 2 species occurring exclusively on healthy, and some diseases occurring exclusively on the diseased tissues. What this also means is that we can now confirm that this also occur as endophytes, but we still need to understand what is their association with the disease that we see in the field. And to understand this, we did a pest trial on Bowman seedlings, where we inoculated the seedlings with all the species that we obtained from my study. And from the figure on my right, your left, you'll see that the seedlings were able to maintain a healthy status where we did not see any tip dieback symptoms or lesions forming. However, after we drastically reduced the amount of water that we were watering for the seedlings, we saw that they started to have the typical dieback symptoms that we see in the field, and also some lesions started to form, confirming their pathogenicity on macadamia. What we also know is that they have been found to cause severe mortality in Australia after an El Nino effect occurred, and also on pistachios, where it caused an entire demise of the orchard after a cyclone. What we have also observed in KZN after the flood is that the disease incidence also, or severity of the disease also increased after the flooding. So what we wanted to understand is the influence of this disease on the, of water stress on this disease. So for this, we are still busy with this trial, but we will be looking at 4 of the macadamia cultivars, A4, N2, 816, and the 695, where we will be measuring stomatal conductance and water potential, looking at both the flooding and the drought stress. This will be done under Dr. Nikki Taylor, who is a plant physiologist at the University of Pretoria. Now that we know what is happening with the branch dieback, we also wanted to see what is actually happening with the other tissues in the macadamia canopy. It has been reported as causing brown leaf blight in China and also causing husk rot in China as well, but we don't know what is actually happening in South Africa. So then we did a very good study looking at all the tissues in the macadamia canopy, where we sampled both healthy and diseased as well, and we saw that some species were found occurring also on all the tissues sampled, while some were also found exclusively. What is important, though, to notice is that, yes, we do find it on all the tissues that we sampled. However, it is important to note that it was more frequently isolated from the leaves, from the diseased leaves, in comparison to racemes and nuts, where we found already known pathogens on nuts such as diapothi and on racemes, as Gerda already mentioned, Neopistolothiopsis. Yes, now that we know that we have it on other tissues in the macadamia canopy, is it really causing the disease that China has reported on these tissues? So for this, we also did a pathogenicity trial, also on Bowman's, and we can actually confirm that from our pathogenicity trial that they are able to cause diseases. However, based on the figure that I just showed previously, we believe that it is going to be more of a problem on the leaves than it is going to be on the nuts and the flowers, based on the frequency that we obtained. Now, what does this mean for the industry? Well, now we can actually confirm that the Botrytis furiaceae is causing multiple diseases in the canopy, and it will be very interesting to see whether the disease is more severe under flood or under drought. So it's going to be a very interesting study. Thank you.
Morning, my name is Dee. I'm the one working on husk rot. So in our study, what we were aiming to understand is improve our understanding of the causal agent and then improve our understanding of the biology of those causal agents. Cool, so we did 2 consecutive seasons of sampling, and this is what we found. So as expected, we found PHR or Phomopsis husk rot caused by diapothes, so that's that black shiny lesion. And then we also found anthracnose husk rot caused by Colotoxicum, which under optimal conditions will give you those nice rings of orange fruiting bodies on your lesion. So we looked a little bit deeper into the identities and found 3 main species of Colotoxicum that were causing problems all over. But for the diapothes, we found multiple species. This can be a problem because if you have a high diversity of species, there's more chance you're going to get variation in your disease. And then we found a 3rd pathogen, also causing some husk rot. We found 3 species. So Colournectria husk rot we found in Limpopo and Mpumalanga thus far is quite new. And as you can see in the picture, it's got this white mycelia or fungal growth on top of the black lesion, which is sort of the normal husk rot symptom. And when we did our path trials, we again saw this nice white mycelial growth on the surface, which was nice, quite a nice field indicator. Oh, sorry, that's all mushed. So when we were doing our sampling, we did an early husk rot sampling and a late season husk rot sampling. On the early seasons, we found PHR, so that's diapothe, in much higher numbers than AHR. However, when we went back to those same orchards later on in the season and sampled again husk rot, we found that the Colotoxicums, or the AHR, was actually much higher than the PHR. AHR and PHR we found in the same orchards on the same tree, sometimes even on the same nut, so they're kind of working together. So then we did some biological studies. We did a temperature growth study. As you can see, at the higher temperatures, Colotoxicum is actually growing much better at 30 degrees compared to the diapothes. So this very nicely explains why we're seeing that switch from having PHR early in the season and AHR later in the season. So we looked at this same study for all 3 of the fungal groups that we're working with. As you can see, that Colournectria group seems to be growing slower. Don't let that fool you. Colournectria is a very well-known pathogen. In China, they have also reported some Colournectria causing husk rot. They reported 50% yield loss with severe infection and quite a quick and rapid growth. So we need to keep an eye on this one. It's not too extensive just yet, but yeah, we need to please keep an eye out for it. So now that we know that there's difference in distribution, there's these 3 fungal groups, the next question is, does that make a difference for your management? So our honors student, Simpo, has been working on this problem. She's been doing some in vitro fungicide assays, just checking out stuff. And so far, there's no difference between PHR and AHR, which is great, so keep going. And then for Calanectria, currently we're seeing low levels of efficacy for the currently registered fungicides. So that just means it's not quite working as well as it should in the lab. So we would really love to keep monitoring this disease. So if you do see this in your orchard, please do send it into the FABI diagnostic clinic. We would love that. It would be great. Yeah, and that's my story. Thank you so much.
Good morning, everyone. My name is Ashley, and I'm doing my PhD on Macadamia nut borer. During my master's, we wanted to know which species of nut borer was responsible for causing the majority of damage to macadamia nuts. So we conducted a survey from 2017 to 2019 throughout the 3 main growing provinces, where we collected and identified larvae from inside macadamia nuts. What we found was that macadamia nut borer was by far the most dominant species. It comprised over 95% of the around 1,000 larvae that we collected during the survey, and during this time we only found 5 FCM larvae. We also wanted to test the specificity of the pheromone traps, so to do that we identified the moths on both the MNB and FCM targeted sticky traps for 2 consecutive seasons in a farm in Mpumalanga. What we found was that both species can be found on both traps. So in terms of the MNB traps, we did mostly find MNB on them, as you can see indicated in red. We did also find a few FCM. More significantly, however, was that on the FCM targeted traps, we actually picked up more MNB than we did FCM. So this is obviously misleading because a high moth count on your FCM traps doesn't necessarily mean that you have a high number of FCM in your orchard. All right, chemosensation, it essentially refers to the senses of smell and taste. These senses are important in terms of finding host plant material, oviposition sites, as well as suitable mates, amongst other things. A common example of where chemosensation has been exploited for pest management purposes is the use of sex pheromones. Sex pheromones are highly species-specific. They are comprised of major and minor components, and these components need to be present in a particular ratio in order to be effective. Sex pheromones have huge potential when it comes to effective and eco-friendly control. However, we need up-to-date research prior to this, particularly when you consider how quickly chemical communication in moths can evolve. During my PhD, we aim to characterize the sex pheromone of MNB, and we also want to identify the genes that are involved in determining what the insect can smell and taste. For this, we will use traditional chemical ecology approaches, largely employing gas chromatography, and we'll also do a reverse approach where we will sequence the genome and the antennal transcriptome. The genome and the transcriptome will be really useful in terms of what information we can get from that data, and it will also create new leads for novel or future controls. So far, we have confirmed the major component of the sex pheromone as Z8-dodecanol acetate, and we have sequenced the genome, and we have a genome completeness score, or a BUSCO score, of 98%. This season, we are hoping to identify the minor component of the sex pheromone and sequence the antennal transcriptome. Lastly, we would like to test different trap types with various volatile blends with the aim of improving moth capture and accuracy. Thank you for listening.
Okay, good morning everybody. My name is Elisa Par, and I will give you an update of our stingbug research that we are conducting in our group. So as we all know, controlling stingbugs population can be very complex and difficult, and developing an IPM program, it's a very long process which require a good understanding of the life cycle and biological traits of your targeted species. And in our case, the 2-spotted stingbugs, or Baticolia distincta, and like as other Pentatomidae species, these bugs has a specific different life stages which consist of an egg, nymphs, and adult stages. And as you can see on this figure, there is different biological traits which are involved in its different life stages, and basically we can target those traits for pest management. And today I will talk more specifically about the genetic, pheromone, physiology, and gut symbiont on these bugs. So the first question that we had when I started my PhD was, what is the genetic diversity of this pest in South Africa? And if you think about it, it's a very important question from a pest management perspective, because a small genetic difference can really impact the efficiency of your pest management program. And what we found in our study, we found that stink bugs from the 3 main macadamia production regions in South Africa are not genetically different from each other. And in another term, there is one species in South Africa at the moment, which is a good news for the pest management. So the second aspect I'm looking at, it's a stink bug's pheromones. So stink bugs are very well known to produce a large variety of compounds, where some of them are really interesting for pest management. And in my PhD, I specifically characterize and identify the alarm pheromone, and we found that 3 compounds are responsible of the alarming effect when the stink bugs release this pheromone. But I'm also looking at other types of pheromones, such as the aggregation pheromone, which plays a massive impact in the life cycle of the stink bugs. And we are currently busy conducting some field trials with different traps and pheromones. And hopefully we will have good results in the future. And I'm also looking at the physiological and biological effects that insecticides can have on the species in terms of acute toxicity first. As you can see, various products can have like different acute toxicity, but we're also looking at what are the sublethal effects that we can see when the insects are exposed to the insecticides in terms of dispersion. I'm really sorry, I don't know what's going on with the PowerPoint. And in terms of other biological parameters, such as the fecundity, hatching rate, developmental duration, or adult longevity. And hopefully those results will help us to understand what is going on in the field with these insects. And finally, we are working on the stink bugs gut content, and here I'm talking on the behalf of Dr. Gerda Fourie and Tembeka, who are working on this aspect. So the first aspect was to identify what the stink bugs can eat, so identify the plant material in their gut content, and hopefully we will be able to identify alternative plant hosts in the future for pest management. And the last aspect is to look at the gut symbiont. So it is well known that the gut symbiont in stink bugs is very important. The female, when they lay the eggs, they're gonna disperse a gut symbiont on the surface of the eggs, and when the nymph hatch, they're gonna acquire this symbiont. And this symbiont is crucial for stink bugs to live, and if you disrupt or remove this symbiont from the egg capsule, you can kill a population. So it's a new way for pest management, and here we're gonna try to repeat what it has been studied on the brown marmoset stink bugs in the US on our South African species. So on the behalf of my team, we would like to thank you for your support and supporting our research, and we really hope that our funding will improve the macadamia IPM programs in the future. So thank you very much.